Hello, my name's Ebony. I am the mother of Orion and Osiris. They are 5 and 7. We live in Ohio, and I have the privilege of introducing Dr. Mackenzie, our next speaker, Dr. Mackenzie. He is a clinician scientist in the University of Rochester, focusing on gene-based therapy in the 4 neurological diseases affecting children. He is a board-certified pediatric neurologist and completed his fellowship in neuromuscular medicine prior to joining the facility at Rochester in 2021. He is interested in improving and understanding the underlying mechanisms and neurological manifestations in the TANGO2-related disorder as we move forward with the treatments in the future. Dr. Mackenzie is a member of the TANGO2 Foundation Committee and is involved in several projects, including the addition of adding TANGO2 to commercial gene therapy panels, determining the genetic frequencies of the pathogenic variant in TANGO2 in the population scale, and involving more specific antibodies against the TANGO2 protein. Please welcome Dr. Sam Mackenzie.
All right, this is great. Thank you for, oh shoot, that's a bad way to start. 1%, 1% better. All right. I'm just gonna say that every time I make a mistake, just 1%. It really is a pleasure to be here. My family and I were on It's a Small World, and I feel like that's an apt analogy for this community. Strangely enough, I was in residency at University of Michigan. I worked with Josh Meisner, who's my co-intern, and I think David Harris back here was he's probably in his first year of faculty on the epilepsy team at Michigan. And since then, I've met so many of you over the years, and we've done a lot of Zooms, and this is the first in-person meeting. Amanda and Jordan and countless other people here. So this has been a real delight for me just to put real faces to Zoom faces, we'll say. So I'm a neurologist, as Ebony said, so I'm a pediatric neurologist. I focus in neuromuscular medicine. And if anybody was present for the virtual meeting we had in the fall, a lot of what I'm interested in revolves around gene therapy. And I'm not going to talk a lot about gene therapy today, but I do think, that this is a disease that is almost perfect for it. If you look at the criteria that we need for a viable gene therapy disease, you have to have a fairly small gene that fits into a virus. TANGO2 is a small gene that fits into a virus. The symptoms have to be severe. We know, from our video and from our own experiences that TANGO2 unfortunately does result in early death in a percentage of patients. And it's a terrible disorder, and it's something that I think is a perfect candidate, and that's really a focus of mine, is kind of helping us get there. So I'm not going to talk about the gene therapy. I am going to talk about some steps that I think we are primed to take in the next months to years, and hopefully put out sort of a rallying cry. In the context of all that you do, I feel bad asking for more, but I think there are some things that we can do and be strategic about doing that are going to move us forward. So I'm going to continue my Disney analogy. This is our family at Epcot yesterday, and we did not eat all these foods, but these foods are apparently available, I'm told, for a price if you go around Epcot. So this talk is going to be a little bit like that, a little bit of sampling, and Dr. Lay did a phenomenal job of setting the foundation for this condition. So I'm going to try to build a little bit on what she talked about in terms of the genetic basis of this disease, what it might be doing at a protein level, sort of teeing up some talks that you're going to hear later, and also thinking about the most important part of this, which is the patient or the kid, and how we can help more kids that aren't at this conference. And that's kind of what keeps me up at night. Unfortunately, so in contrast to the last slide, this was my Amazon Fresh order, and they take their name very seriously at Amazon Fresh, because I think somebody just like clipped these off of a tree in the rainforest, and they are still very green. So that's kind of an apt analogy for where we're at with research. So these bananas will be good maybe next month, and so that's a little bit where we're at with our research. I think we're going to get there. We're doing the right things. It's, going to take a little bit of patience, which is something that's hard to exert when we know what's happening with this condition, but we are moving forward. All right, so just to refresh everybody after the break, so we talked about, Dr. Lay talked about the TANGO2 gene and what genes are, and I think everybody knows that. And by the way, I don't know if this has been mentioned, but in the back of your program, there is a glossary of terms. So if you hear some buzzword that somebody mentions, all of that is there and accessible to you. And if it's not, you can absolutely submit questions using the QR code. But genes are basically instructions. It's an instruction manual for making protein. And there's an intermediate step in between the gene and the protein, and that's called RNA, and I'll show you a little bit about what that is. And genes are kind of wound up really tightly within chromosomes, and we all have 23 chromosomes. We think about it as like, oh, we get a set from our mom and a set from our dad, but they kind of mix and mingle in a way that your chromosomes are not exactly identical to one of your parents. They're about 50% of your parents' DNA. And each chromosome contains or each set of chromosomes contains a full package of DNA. So we have 2 genes basically for all of our chromosomes. The exception being the sex chromosomes with X and Y, but we don't need to get into that. So just in terms of how this DNA or this code is expressed, I've talked about this like a cookbook in the past. So it's kind of the instruction manual for making a protein. There's really just 4 letters in the alphabet of our DNA, and those 4 letters are A, T, G, and C. And you can see there on the top, the DNA has a series of these A's and T's and G's and C's. And this is kind of a wild concept, but only about 1% of all of our DNA actually encodes protein, and the rest of it is important for things we probably don't know all of the reasons for, but it kind of regulates which of those genes gets turned on and expressed. So which of those cookbooks are we pulling off the shelf to, make a cake or make a loaf of bread? So those can happen at different times in different cells and different tissues. But that 1% is definitely important, and we know that. So what happens with, on the way to making a protein, the DNA turns into RNA. So little enzymes come through and they're reading that code, and that code gets turned into messenger RNA. So there's the M there in front of RNA. And the only thing that's really different about RNA is that we now have U where that T used to be. Other than that, it's pretty similar. And this is where it really gets cool. So with RNA, the key number is 3. So for every 3 chunks of RNA, that's going to correspond to something called an amino acid. And if you see here, the first 3 here are AUG, so that tells something called the ribosome that's reading, now it's reading the RNA. The ribosome sees that AUG and says, oh, I need to start making protein. And what it does is it starts with an amino acid called methionine, which is the MET at the bottom. And then it looks for the next chunk of 3, right? And that AUC in this case is going to say, oh, let's make isoleucine. That isoleucine now attaches to the methionine. And this continues kind of in this building block progression. So you can see here, you can already probably start to appreciate, if I just took out one of these, right, and I'll show you some examples in the next slide, you could really cause some problems, because the ribosome eventually, in this example, next one is a serine, but then it sees UAA, and that's a specific code that tells that ribosome, let's stop here. And so our building blocks of amino acids get stopped, and whatever protein was made or wasn't made gets sent out there. And if it's not a functional protein, our body's really smart and it just chews it up. It gets degraded inside the cell. So this is why certain mutations, as the old term, or variants within a gene could cause disease or could not. And so this is my 9-year-old son, and I did not get his permission to put this picture up. So this is him playing in the outfield, baseball ready, as his coach likes to say. And I'm going to use a example here on the top. And I told you there's 4 letters in the genetic alphabet. I'm just going to make this a little bit more user-friendly. So we have a sentence here: the boy may hit and run. If you read that, you could say that's totally a sensible statement and makes sense to me. We'll call that a viable protein. Okay. Now, if I have 22q11.2 deletion syndrome, right, I might be missing that whole chunk of my genome. And if I just crossed all of those pieces of DNA out, I don't have any sentence at all. And that's kind of what we see here in the second sentence, where I just crossed it out. So nothing there, right? Third example here: the boy may, and now you see hit and are crossed out. So the boy may run. Makes sense, right? So we could consider that a viable protein, and it's going to do the same job. It gets the message across. Maybe it doesn't get the whole message across, but it's something that might have some function or might have some efficacy in the cell as a protein. The next one, so if we swap out a letter, so we call this a substitution of a base pair. I took out the B and I put in a T. The toy may hit and run. Also kind of makes sense, sort of, maybe not completely, but it's a sentence, so that protein might have some function inside the cell. Now, if I swap it out for another letter that maybe doesn't make sense, the Zoe may hit and run. Nonsensical protein. That protein's going to be swimming around the cell and might even be damaging, because now it's got some new function that doesn't make any sense. We can also have deletions. So I crossed out in the second to last line the B, and this is where we come back to that magic number 3, right? So if I take out the B, that ribosome that's reading the messenger RNA is going to get to and then it's looking for the next set of 3. So now I'm not even going to try to pronounce that, but that's clearly going to be a nonsense sentence. That's what we call that, or nonsense transcript. And eventually down the line, somewhere in that sentence, that ribosome is going to get to that stop signal and tell itself, okay, we're done. And that whatever was printed out just kind of gets chewed up by the cell, no protein. And same thing here, I've added a G, right? So this is called an insertion. And when we do that, you can also get a shift. So you might have a deletion of 3 and you get away with it, because again, that ribosome is reading in sets of 3, or an insertion of 3. And those are cases we might not even detect because, again, they don't cause disease. So there's a lot of variants that can happen inside the gene. A variant doesn't necessarily cause disease. It's not necessarily pathogenic. You guys have all probably been through this in some version with your genetic counselor at some point along the way. All right, so again, I'm not going to belabor this, but we all know the term autosomal recessive, or you might have heard that before. So that just, again, means you need 2 copies of a misspelling in the gene in order to cause disease. And you could see here, maybe the orange one might be a variant in TANGO2 that causes disease, the green being what we call the wild type, or the normal version. And child 1 in this case gets 2 full copies without the variant that causes disease. Child 2 in this case would have 2 pathogenic variants and have TANGO2-related disorder. And Dr. Lay also kind of touched on this. So this was data, these were data from the Broad Institute. Really cool collaboration, and shout out to SEMA and a lot of others that made this happen, and the Morrises. But I think this was a huge win for this disease because it's firepower that our community can take to other resource organizations and say, look, cold hard facts, this is the expected prevalence, 6,000 cases worldwide. We've diagnosed, maybe 140. And that just speaks to the work that we have to do to really expand the diagnostic testing. And that's going to be kind of a theme of this talk. So all populations across the board, top column, we could see maybe 1 in 530. But if you look down in some of these, in that third column, you see the non-Finnish European population, and that's where most commonly that exon 3 to 9 deletion is seen. We're up to 1 in 321 individuals. So we're approaching 1 in 100,000, which is not ultra rare. I mean, it is by our numbers, but conceptually, there's probably a lot of people out there that are missing this diagnosis. And then the other thing just to draw your attention to is that other line, and again, this speaks to a bigger problem in that our population databases are largely data sets full of European ancestry. So we know that there's the G154R variant in, did I get the number right? It's not 154. It is 154. Variant in the Texas and Mexican communities. And that's not super populated in the NOMAD database where we draw a lot of these numbers from. So we have a lot of work to do in terms of being inclusive about diagnostic testing. This was a quick little picture from our paper a couple of years ago where we just looked at all the cases that had been described, and I think at this time it was 76 patients with TANGO2. These are diagnosed cases in the published literature. And if you just kind of walk across the gene, that's the boxes at the bottom. So each one of those numbers is an exon. Again, those are the coding regions of the gene. And we can see that there's variants in almost every exon that we've found. And the asterisks, those little stars inside the circles, denote the cases where one of those letters is just swapped out. So that's called the Missense Variant. And I've harped on this. If you saw my talk in the fall, I think this is a really key piece of information as we're still in the learning stages of this condition, because that gives us a little bit of insight into what parts of the protein are the really important parts, right? If there's a certain amino acid at one of those positions where those asterisks are, and now we're changing that amino acid and the rest of the protein stays the same, we know that that amino acid is doing something important in terms of the protein shape or the protein function. So I've kind of been very interested in figuring out exactly what's going on at those sites. You can see on the bottom too, just for kind of a sense of scale, the 3 to 9 deletion being at the bottom, which is populated, it's about 45 to 50% of all alleles that we know of in diagnosed cases, and then some of the other multi-exon deletions. All right, so I'm going to come back to the protein because this is gene to child conversation, but I'm going to come back to the protein in a bit, and I just want to pause and talk about kind of the call to arms piece. So it's in the program, and I think have every right to celebrate the fact that TANGO2 got added to a lot of these commercial gene panels. I just want to do a quick, if people are willing to share, and you don't have to, could I see a show of hands for how your child was diagnosed? So I'm going to start. How about a gene panel? Let's say epilepsy gene panel. Other gene panel. One. Do you mind sharing? You don't have to.
I don't know what the gene panel was, but it went over to Netherlands and they did a full metabolic screening.
So probably a metabolic of some kind. Okay. How about whole exome sequencing? That's almost everybody in here. And how about a previously diagnosed child? No. Okay. Anything that I missed? Anybody with whole genome sequencing? A few people with genome. Okay. So that's great because we're moving into an era where exome and genome are becoming cheaper and faster and easier to get in the hospital. But I don't want to wait until the kid is in crisis in the hospital to get this diagnosis, right? And I don't think anybody in this room wants to, which is why I think it's really important that we keep the pedal on the gas with these panels. So there's a few things. One, we haven't won the war on the commercial gene panels. There's still a lot of companies out there and a lot of symptoms. So if I'm a neurologist and I see a child with ataxia, I'm actually not as smart as I might make myself out to be. I just go back in my room and I type out ataxia gene panel, right? And that's literally how these things get sent. And that gets whisked off to some company and they take the DNA and run it. And they look for all of the genes that might cause ataxia. And I would just say it today. I think this foundation could use a diagnostics committee because this is work that we need to check in on every 6 months or so to make sure we're still making progress. There was a lot of companies when we reached out, they said, oh yeah, we'll look into it. Well, we need to kind of keep them on that because it does take advocacy from patient communities and we've done it before. I mentioned the population genomic databases. This is a bigger problem, but I think people are aware of it and working on it. We need to educate a lot more clinicians about all the symptoms with this disease, and certainly look for the disease more in high-risk groups. And I have a few slides on that. And then the last thing, we talked about spells, right? So spells can be one of the first presenting signs, and they're confusing to pediatricians. They're even confusing to neurologists. So I'm going to mention a little bit of work that we're planning to do as part of the kind of an extension of the natural history study to put better terminology on what those are, because again, that's going to lead to more kids getting diagnosed. So Erica mentioned that this is the zip code for the TANGO2 gene, 22q11.21. So if you are already missing, if you're already missing a chunk of your 22nd chromosome, your risk for having TANGO2 disease could be as many as 1 in 321 if you're a non-Finnish European population, which is insanely high, right? That is a really high number. And you could make a strong case that we should be sequencing all of these kids with DiGeorge syndrome to see if they have a variant on the opposite, on the other allele. But we did a small project where, when I was at Nationwide, there was a 22q clinic, and we went through the charts of all of these kids, and we looked for symptoms. So we kind of did this in a little bit of a priori prediction of what symptoms we might see. So here's a Venn diagram. The 22q population might have congenital heart disease, commonly tetralogy of Fallot, cleft palate, immunodeficiency, and seizures, developmental delay, and hypothyroidism. And if you look at the TANGO2 circle on the right, some of the things that are unique to TANGO2 might be the metabolic crises, certainly rhabdomyolysis, the tachyarrhythmias, cardiomyopathy, and movement disorders. And so we wanted to see if we could establish some sort of criteria, or at least test these criteria to see if we could identify patients with TANGO2 within the DiGeorge community. And so we kind of split them down. Those ones that were unique to TANGO2 we counted as major criteria. The ones that were overlapping we put as minor criteria, and we just kind of flagged kids. So if you had one major criteria or two minor criteria, we reviewed you as a group. So there's kind of a few steps. So we started with 293 kids in the database. One of them we didn't have any data in the EMR. A few on review didn't actually have 22q11 deletion syndrome. It was just misrecorded. So we did a full chart review on 255 patients, 9 of which had passed away, and we flagged 140 of them had at least one symptom. And so by the time we did a consensus review, we kind of looked at the criteria and we said, well, this doesn't really fit, or this person had metabolic crisis, but they were also really sick because they just had cardiac surgery for their tetralogy of Fallot. We ended up recommending sequencing for 7, and I still don't have the data for those 7 because it's been more of a slog than we anticipated reaching out to those families and getting testing done. But we're hopeful. And this is just to point out, I think we could also very feasibly put together those criteria as a community and be in touch with the 22q community, because these are kids that are at very high risk. This is a slide just to kind of show some of our disagreement. So there's just 2 reviewers that did the full chart review on every patient. And we had disagreement, even as clinicians. There's me, a child neurologist familiar with this condition, and then another physician who is a pediatric geneticist. And without having clear definitions of what these things are going in, we disagreed on kind of what constituted spasticity. And again, you're just reviewing a chart, but this is probably more detailed than you need. I just think we need to be give a coherent set of criteria to this community for when it's appropriate to test, and certainly make it known, because I still think that there's a lot of geneticists that see 22q patients that don't know about TANGO2. Here's just one young woman who we identified on chart review that we're going to recommend testing for. Does she have TANGO2-related disorder? I don't know, but she did have intermittent hypoglycemia as a child and had an episode of rhabdomyolysis at 20. I think that right there, if you're seeing this patient in clinic, it warrants testing. So another project I'm very excited about, and the Taggarts are helping with this. There's opportunities at this conference to sign up. It just requires reconsenting as part of the natural history study, even if you're part of it. What we're hoping to do is review videos of kids that have had these spells or other strange episodes that have perplexed your pediatricians or maybe even your child neurologists, and just review those and curate them and put a name to them, because again, that terminology is really critical. If I say, oh, that looks kind of weird, I don't know what that neck tilt is, did that go away? Oh, I guess we'll just see if it happens again. That's a very different encounter than if your physician says, oh, that looks like torticollis, that's really strange, and intermittent torticollis, let's see what causes that. That might create a diagnosis for that kid, right? So we need to be clearer about our words as families and certainly as physicians, because words matter and words lead to testing. So what we want to do is just have, all it takes is video. It can be old video, and we're going to upload it. And there's 3 movement disorder specialists where I work, which is weird. That doesn't happen that often. But Jen Vermillion, Angela Hewitt, and Jonathan Mink, all pediatric movement disorder specialists. We're just going to sit down in a room together, and it's going to be really ugly. We're just going to fight it out until we label all of these videos and hopefully put something out there in the literature that says, here's what we see with this disease, because there's a lot that's been described. I just don't think it's been done systematically. So let's go back a little bit to the protein, and I just want to tease some talks that are coming. I'm still kind of getting my lab up and running, so we haven't done as much work as I would like to in this area. Erica mentioned this too. So what do we think the protein does? We really don't know. We think it's in the cytosol, which is kind of the fluid compartment of the cell. It kind of goes everywhere. It might move around to different cell compartments, such as the mitochondria. And we'll hear from Lena Gonzalez later, I think just after me, that when we take away TANGO2, mitochondria get unhappy in certain situations. We think it's also important for protein trafficking. I was totally a protein trafficking nihilist a few months ago, but now I'm back on the protein trafficking train. I think it's got to do something because these symptoms are just too weird and we don't see them in other mitochondrial diseases. So I don't think that's the whole story. We'll hear from Dr. Sasher about what it might be doing with other proteins, so it could be interacting to exert its effect. But we know it's important because a lot of species have TANGO2 or some version thereof. So we call that conserved. It's very highly conserved across species, which probably speaks to its importance. Again, looking back to the Missense variants, this is just a little representation of a computer's prediction of TANGO2. And all those little red spots are places where we know that there's missense variants that might cause disease. So those amino acids are doing something critical in terms of keeping that protein structure the same, or maybe even doing something functionally. The little chart on the right is just a representation of each of these amino acids in different species, and you can see basically every species has a C in that second position. So that's a cysteine. And that's a amino acid I'm really interested in as well. You know, we've speculated on what this could mean in terms of function, and it's definitely in the green banana stage. So I don't want to launch into too much detail, but we think you know, there might be some activity that's conferred by that cysteine and maybe some of the amino acids around it. At Rochester, we also have a group of basic science collaborators. This is Keith Nurki on the left and Andrew Wachtovich on the right, who are experts in mitochondria and in C. elegans, which is a worm. And worms are super cool because you can look at them under the microscope and you can squish them up, and you know they don't really seem to mind. If you look at the worm. The worm actually has two genes that kind of look like TANGO2, so it probably duplicated somewhere. But all of those sites where we see missense variants, those are the green things. So those are the same exact amino acid in the worm, even though the overall identity is only about 25%. The yellow stripes are where those amino acids are a little bit different, but they're functionally pretty similar. So as of yet, we haven't been able to get much out of the worm. But we're still hopeful. So yeah, this has just been a joy. We just sent in a grant with Christina Mayaki, Lily Zhang, Michael Sasher, Lena Gonzalez. So you know it can seem like we're all in silos as researchers, but this was a great experience. Submitting this Chan Zuckerberg grant, we're crossing our fingers that'll be back in October. But it's, I think, again, room for some hope that we're moving in the right direction, and I think collaborative research is the only way we're going to get to the finish line with this condition. I presented this slide in last fall, and again, I think this is kind of how I think about the finish line and how we get there. And these things do not happen in sequence; they happen in parallel. So it's not a linear road necessarily. But we've had a lot of these retrospective studies. The natural history study is 4 years in. We're starting to learn about disease mechanisms at the same time. We're developing animal models. You're going to hear about some of those. And I think the coolest thing about this conference, and I'm not going to spoil it, but there's some data out there on therapeutic intervention. And I just think that's going to be one of the biggest things we remember from this conference. Hopefully, in the not-so-distant future, we'll move into clinical trials and have a treatment for these, whether that's gene therapy or small molecule or something else. And you know, then we'll still have more work to do to make sure that we're not seeing long-term effects even on treatment. So, lot of work to do. It seems overwhelming. I just think that. Again, we're making good progress. There's a lot of reasons for hope. We need to keep building the team, and I say that in terms of the patient community, and that's just diagnosing more kids, and also building our research team. That's what that money goes for when you're out there, you know, slaving away, which is, I recognize, is doubly unfair as a rare disease parent to also have to raise money for your kid's rare disease. It's not fair. It's the way it is right now. I'm right there with you trying to raise that money, but we need to get more researchers involved. get more diversity of thought. And then we need to think about the low-hanging fruit. So that's advocacy efforts like, keeping the pedal to the metal on the gene panels and looking at these high-risk groups like the 22q population. This is just our Rochester dinner with Amy and Stan and Katie. And there's Amy and Katie. These are cans, they're not garbage. So we had a can drive, and it's been a lot of fun. Very rewarding work to work with all of you, and yeah, it's been a pleasure. So thanks so much for your attention.